From Microbe TV, this is Immune, episode number 62, recorded on November 22nd, 2022. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back. Good to see everybody.
You can get a lot of snow up there. That was Buffalo, right?
No, we're not Buffalo. 3 hours away. See, everybody's like, well, that's just New York. It's upstate. They're all the same right now. It's quite isolated. We did get some snow, but only a dusting. So.
So Cedric was on Twiv on Friday. He said you got a dusting there. Yeah.
Yep.
Also joining us from Cleveland, Ohio, Steph Langle.
Hi. Great to see everyone. Last time we saw each other, we were in a tropical climate.
Yes.
I'm now in a much different climate. My first podcast at Case Western Reserve University. Very thrilled to be here and honored to be with a guest who we'll get to introduce in a bit.
Steph, this is the first of many podcasts from Case, right?
Listen, moving a lab is not the most fun thing. So I'm going to stick around for a while, I think. Yeah.
All right. Very good. And from Madison, New Jersey, Brienne Barker.
Hi. Great to be here. Sad that I'm seeing you over a computer instead of sitting together in a tropical location. Yes. But I'm excited about our conversation.
Yeah, those times when we're together physically are rare. You have to just enjoy them when they do happen. That was a nice opportunity.
Yes. It was.
Our guest today is the Presidential Professor of Immunology and Professor of Pathobiology, University of Pennsylvania School of Veterinary Medicine, De'Broski Herbert. Welcome to Immune.
Oh, thank you so much for having me today. I'm pretty excited about this.
Yeah, we're looking forward to chatting about you and your career and your work. And you're currently at the university today, or are you at home? I don't remember what you said before.
Yeah, unfortunately, today is a home working day due to reasons, causes outside of my control. I would otherwise be in the office.
These things happen. Now, Cindy, you had originally reached out to De'Broski. Is that correct?
I did. I pitched this because I saw De'Broski at a meeting recently. We were at a conference called the New York's Upstate Immunology Conference, which is just a lovely conference. De'Broski was a keynote speaker there. And I think you can agree that is really a wonderful community, a really great conference. And I got to hear his science, which was just outstanding. But he also talked a lot about his story, and we had a really nice workshop with him too. He has a lot of interests, and so I thought it would be really fun to have him on to talk about this.
This was an in-person meeting? Or was it?
It was. Cool. Yeah, right before I came to the Society for Leukocyte Biology conference. It was the week before that.
And where was that held?
It's in Cooperstown, New York. Do you know what Cooperstown is famous for?
Yeah, the Baseball Hall of Fame.
Baseball Hall of Fame.
Know this, right? Yeah, Baseball Hall of Fame. Yep.
Is it baseball or not basketball? That's in Ohio, right?
It's baseball Hall of Fame. Yep. Mm-hmm.
Football Hall of Fame is in Canton, Ohio, which is actually where I grew up. So
that's what I'm thinking about. So where's the Basketball Hall of Fame? I don't
Springfield.
remember.
No idea.
Springfield.
Yeah.
Yeah, right.
Yeah, that's right.
So what is that, a Jeopardy question, Brienne?
It sounds like it.
Do you know that Brienne was on Jeopardy recently, De'Broski? Did you know that?
Wow, I did not know that.
Yeah, and she won.
Mm-hmm. What? Yeah. Really?
Yeah, she did. Yes, she did.
Pretty cool.
Pretty awesome.
She's a star.
That's pretty impressive. Yeah.
So
cool.
she is our keeper of all things trivia.
Everything. You got a question?
Immunology and otherwise. Yes.
I've never met a walking encyclopedia before.
That's great. Walking encyclopedia. Okay. So, De'Broski, we'd like to start by hearing all about your career, starting from the very beginning when you were a little guy. Where was that?
Wow. I love telling this story because it defines so much of who I am and my passion. So I grew up in South Mississippi in a place called Hattiesburg. Mm-hmm. It's about 100 miles from the Gulf Coast. And, previously, of course, before the Rockefeller Foundation set in, hookworms were endemic in places like Mississippi and Alabama, Georgia. And actually, there's still a problem along the Appalachia regions of the US. But as a young child, my great-grandmother, who served a very key role in my life, in my formative years, routinely warned me to not go outside without my shoes on. So as you said, a little guy. I was a little guy that, 6, 7 years old, and it's, 85 degrees humidity and 90 degrees temperature in South Mississippi during the summer months, and shoes just seemed to be punishment, and but she insisted, you must not go outside with your shoes on. And that always just stuck in my head. She wouldn't, of course, explain why. My great-grandmother, Addie Mae Jackson, was born in 1903. She's from the generation where you do not question your elders. Okay, so fast forward to sophomore year at Xavier University in New Orleans.
Oh, wow.
This would be probably around 1992. My great-grandmother had passed by this time. I took a parasitology course because I recently switched my major from biology pre-med to microbiology. And this was a strategic move to change my major because there was the MARC fellowship, the Minority Access to Research Careers. And being someone from a low, kind of income background, wow, a full ride to go to school, and all I have to do is this thing called research. Of course, I'll change my major, I'll do whatever. And so I became a microbiology major and took a parasitology course. Well, probably the, second, third week in class, I learned about not only hookworms, but schistosomes. But it was really the learning about hookworms that just blew my mind, because then it took me back to 6 years old. Now I knew what my grandmother was talking about. And I just became fascinated by, here are these organisms living in the soil. They're able to directly enter your body and live within you and feed upon your tissues and stay for decades. I was just completely mesmerized. And there were more examples of this. So all throughout the parasitology realm are organisms that have evolved to live within us for as long as they can. And so then you fast forward a little bit more. I then went on to Thomas Jefferson University at this time as a graduate student and was exposed to immunology. I had taken immunology in undergrad, but, I didn't know that was going to be my career. But as a graduate student, it then dawned on me that combining my interest in parasitology with this emerging field of immunology that, it's arguably only been born in the 70s. The immunology field is not that old in relative terms. That's what I wanted to do. I wanted to combine the latest in scientific understanding about how biology works with my childhood interests, and that drives me today.
So when you were a little guy, were you interested in science?
Oh yes, it was always my favorite course. Actually, in 5th grade, I wish I knew where this poster board was, but the kind of, what do you want to be when you grow up? Well, my first. Don't laugh. My first aspiration, my absolutely idol in 5th grade was Jacques Cousteau. So I wanted to be an oceanographer. I thought he was the baddest guy ever.
That is so funny you say that, because I too remember reading a book about Jacques Cousteau, and that name has stuck with me for for years. How funny. That's so funny you say that.
Right. So because I drew myself, in scuba gear, taking pictures, and I was, what are you talking about?
That's awesome.
Right, South Mississippi. Yeah, I want to be an oceanographer. Yeah.
But you had said you were pre-med, right? You wanted to be a doctor?
Initially, yes. Mm-hmm. Xavier University has still probably the top reputation out of all the historically black colleges and universities of placing African Americans into medical schools across the country. So it is a premier undergraduate institution for someone with my demographic. And I thought, well, I like science, and, being a doctor, particularly in the African American community, is something that, your parents and so on and so forth said, yeah, are you going to be a doctor one day? Well, oh, okay, yeah, I'll be a doctor, but I didn't have, let me put it this way. I don't regret it one bit. I think I'll probably be an awful physician. So I think it worked out just right.
Yeah, I think I would agree. I would definitely agree. Can you tell us about your transition from graduate school to faculty, and what was that journey like? What brought you to Penn, and particularly in a veterinary school?
Okay, yeah. I mean, it's kind of a long story.
We're used to
that.
Yeah,
you're used to that. Okay. I will first start off with the not so nice parts, because I think there's a lot more of the great parts than there are the parts that are not so nice. This path has been particularly grueling. Only in these recent years have I really, truly enjoyed all aspects of my career today. As a trainee, being someone, you have to realize that I was in grad school between 95 and I finished my degree at the end of 2000. Okay. There were really no other people who looked like me. And, my experience had mostly been in the South, where, no secret that our country still is under the burden of race relations and so on and so forth. And, for me to have this aspiration, there was a lot of, scrutiny, maybe is the way I would like to put it. I started in a master's program, but I immediately transitioned into the PhD program. And in order to do that, I had to take several exams and, have many interviews and so on and so forth. But even after entering grad school, at that time in the mid-90s, and I don't think it's particular for Philadelphia, to have racially insensitive comments made was commonplace. Actually, I would say a good friend of mine, a person that I worked with a lot during grad school. He was also a grad student, he was an upperclassman. We got along fantastic. We would play chess and he taught me a lot of things. He found it important to point out to me, had I missed this book called The Bell Curve? And that, people like me are thought to be largely intimidated by the basic sciences. And what was I doing? Who did I have the goal to say that, I want to be a PhD in a basic science discipline, not like African American studies, nothing against the humanities, but, there is a considerable difference in the rigor of the training that's entailed and the complexities and so on and so forth. And so I had to endure that. And even if I can go on and on and on, and with many different instances where even after becoming an assistant professor, and I'll come back to how that happened, being invited for talks where the person who invited me would be shocked. Or even I would be in full, suit, shirt and tie and being asked, are you showing up to be the painter? Are you painting? Or who are you and why are you here, basically? Right. That was well into when I was a faculty member. So this path has been very challenging. I think, as Cindy mentioned, I've worked a lot on emotional intelligence because that has been key for me to maintain a high degree of professionalism and communication and so on and so forth, and to constantly focus on, as Maya Angelou would just say, keep your eyes on the prize, right? Don't get distracted and go off on a tangent. Keep your eyes on the prize. And I must say that now here at Penn, I feel like I'm really holding the prize in my hand. Penn is such a wonderful place with all my colleagues and with the students and all of the changes that have come about. And it's a great experience I'm having right now, being accomplished and a little bit of accomplishments, we
would say accomplishments for sure, a lot.
But being recognized and being accepted and being feeling like part of, there's still little challenges. There will always be challenges in life, but I would say it's excellent right now. So I finished when I finished Thomas Jefferson in December 2000. To my core, I was fed up with the U.S. I was just fed up, and I had only considered taking postdoctoral positions outside of the country. So at that time, I wanted to go to Africa because I took a few African studies courses in college and so on and so forth, and read about a lot of people, a lot of scientists, who were of African descent. And, so I had all this, I had kind of a glorification, if you will, of what it would be like to go to Africa. But, there are very few opportunities, and it's mostly through networking. I'll keep talking, but just a minute. That I was considering several positions. I was considering a position in Nice in France. I was very strongly considering going to Switzerland to work at the Basel Institute, which is no longer in existence. Actually, I had secured a position at the Basel Institute, and it coincided with them losing their funding through Merck, or through Roche rather, and that opportunity dried up. And then the fateful day when I had popped the trolley and went from Jefferson over to Penn. I was always coming to Penn as a graduate student. So somewhat, being a professor there is so surreal, and they're on the bulletin board. That was back in the, can we remember when they were actually bulletin boards?
With tax and everything. Yeah.
Right. And I love bulletin boards, right? Because it was just all this rich amount of information. Like you never know what was going to be there. And it was just
and really historical because you keep putting new things over top the other ones, right?
Yes,
you look back and see what happened.
Exactly. And the fateful day when I was over there and I picked up my parasites, and I was on my way back to Jefferson, and there it was. 2 postdoctoral opportunities, Cape Town, South Africa.
Wow.
And there was like about moment where the sunlight comes in and the flowers and the birds, and because I knew at that point that that's what I was always looking for, to go to somewhere in the African continent and do basic science, because of course, I'm a scientist in my core. So, as an aside, I got so desperate to go to Africa. At one point I was considering doing Peace Corps, but that would have been a great experience, but I don't know how much it would have pushed my career forward. So off to Cape Town I went with a one-way ticket. Wow. Very shortly thereafter. I mean, my fellow classmates thought that I had kind of completely lost the plot. Many of them went on to NIH or to really top-notch labs across the country. The immunology program that was there was tremendous. Actually, the 4 other grad students that I was close to, they're all prominent professors at this time. It was like a cohort effect, I think. But that experience changed my whole life. It changed my whole life, and I don't say that lightly. Starting as I got a one-way ticket. I showed up. I had signed off on a house to rent. The exchange rate was something like 12 to 1 to the dollar. So I went from being poor, hungry graduate student to being, upper middle class, And I got married. I married a postdoc who was in that lab. She was South African. We're no longer together, but it was a very rich time in my life. And we published a very key paper. My job was to work on schistosomiasis. And, this is a disease that is a parasitic disease that affects 250 million people worldwide. It's waterborne, transmitted from snails in the water. And my job was to bring that whole model to the lab and leverage that parasite model with some of the existing mouse strains that were in the lab. The lab was headed by Frank Brombacher, who trained with George Kohler, who got the Nobel Prize on hybridoma technology, and Cree-Lox technology. The ability to specifically excise pieces of genomic DNA in a cell lineage controlled manner had just started to take off. And because that technology came out of Germany, out of the Max Planck, but largely, there's a few individuals responsible for that. Frank had inside, so he came from the Max Planck. So he had the inside track on this technology and the mice. So he had all of this in South Africa. We literally had mouse strains that nobody else in the world had. And right about the time when I was starting that postdoc, there had been a few position papers written on perhaps there's this pathway where macrophages would undergo a different form of activation, different from what had been historically described, I think first by Ilya Mechnikov, considered the father of myeloid phagocyte biology. But there was this possibly this whole new paradigm, but nobody had any data. It was just a theory. And the work in that model, that manuscript was the first to formally demonstrate that animals that specifically lacked this pathway could not survive the infection with schistosomiasis. Wow. So it was a life and death phenotype. It was very profound. It wasn't a small little, I need statistics to figure out if this is a biologically important phenomenon. It was like either the mice died or they lived. And that, I still remember getting the data. I was walking down from the lab. That the laboratory was located in the Groote Hospital.
Excellent.
Oh, of course, our trivia expert.
Well, I take students abroad to South Africa each year. And in fact, we spend a day there. Wow. So you can imagine that how majestic it is. Mm-hmm. Cecil Rhodes did a lot, and one of the things he did was build Groote that sits upon a really nice, it's just like you can see over the city, and it's very majestic. And I was walking to my house from the hospital, and I was just lost in thought about what this could possibly mean. And the real specifics of it was I had two strains of animals. I had animals that specifically lacked signaling through key type 2 immune response cytokines, which are molecules that communicate between cells and tell them what to do, but in a T cell specific lineage. So you can think about how important T cells are. Ask anyone with HIV or any host of diseases. T cells are really essential, right, for long-term adaptive, inflammatory and protective immunity. Well, I had animals that lacked this receptor, IL-4 receptor, in the T cell lineage, and I had those that lacked it in the macrophage lineage. And fresh out of immunology PhD, I was like, oh, I know what it's going to be. It's going to certainly be the T cell. The T cells that, when we knock them out, the mice will not do well. There's going to be, very bad outcomes. And macrophages, what do? They don't do much. And it was the exact opposite.
Wow.
It was the exact, actually the T cells, they were better off. So it was a real key finding. And that, I must say, it set my career in to now. I mean, I just was at a talk where someone introduced me as, the guy who, figured out if M2 macrophages were real, and that's in a textbook and all of
that is so
cool. So yeah, it's remarkable, right? And so basically I had a phenomenal time in South Africa. And not only that, but, the country at that time had just become democratic in 1994. So I was there 6 years after becoming a democratic nation. So there was an amazing melting pot feeling. Like I had a host, I think my first year I had in my house, 4 roommates, one from Cuba, one from Angola, one from Denmark, and one from Italy. Wow. Like we were in there all at the same time. It was just everyone, everything. And I was still young back then. So it was, it was fun going to the beach and, doing a lot of great things. But yeah, publishing that paper really set the stage. And so when it was time to go, it was about 4 or 5 years, I was starting to get homesick and, knew I wasn't going to live the rest of my days in South Africa. So I convinced my then wife that, okay, we need to bring our child, back to the U.S. Mm-hmm. Well, I wanted to work with, was arguably one of the fathers of type 2 immunity, Fred Finkelman. I had read his papers all throughout grad school. I knew he was in Cincinnati. I didn't know much about Cincinnati. I didn't have any bias against it. And I was thinking, wow, that would be a nice next step, to be mentored by such a prominent person in the field. And I didn't know how to go about it really. And Frank, being German and being mostly collaborators in Europe, I was like, well, and Fred sent an email to Frank saying, I currently have a position. Do you have anyone who may like to come?
Oh, that's crazy.
Really?
Well, you've had really these moments.
Birds come out.
The birds
sun,
with the sun. Yeah.
The aligning of the stars.
Right. And I was like, well, that's it. That's it. And so that's where it really started to take off for me, because Fred, of course, was card-carrying immunologist, American, knew the federal government system, knew NIH really well, all these things that I couldn't learn in South Africa.
Right.
Right.
And so I started under his tutelage. Then I went from research instructor to research assistant professor, and then to assistant professor tenure track. So I love interacting with trainees today because I'm saying, well, I've kind of held every academic position that you can have. So
I could give you a wide range of advice.
Right. I can talk to you about all of them. And so before I knew it, I mean, by the time, 2010 rolled around, I think it was 2010, I had 2 R01s, I had my own laboratory, I had my own team. It was great, My ex-wife, however, did not really like Cincinnati, being a native Cape Townian. We were earlier before the recording talking about snow, and she had never experienced snow. And so
there's no beaches, unfortunately, in Cincinnati.
No beaches in Cincinnati.
It's a nice place, but no beaches.
Yeah, I loved it. I was having a great time. But then the opportunity came to join the faculty at UCSF in the Division of Experimental Medicine that was formerly led by Dr. Joseph McCune, who was credited with the first generation of humanized mice. Quite a dynamic, phenomenal investigator in person, by the way. But so Mike was building a division that was focused on chronic infectious disease, and he didn't have a helmet person. But more than that, here's another interesting twist. During undergrad, after my sophomore year and after my junior year at Xavier University, I went to UCSF for 2 consecutive summer training research experiences. So I was there as
you had that experience.
I had that experience again, right? I was there as an undergrad. I knew the lay of the land. I love the Bay Area. I mean, it's pretty hard to not love the Bay Area, right? I mean, that's why there are songs and poems written about San Francisco, right? And so if you put those two together, an incredibly beautiful city and the chance to work at a premier research institute in the whole world, I jumped at the opportunity. And so between 2012 and 2016, I was assistant professor, and I did make tenure towards the end of that last year. I made the hurdle, associate professor of immunology at UCSF. Unfortunately, with the price, the cost of living and the cost of doing science and also housing market was just really the linchpin there that just sealed the deal. I knew it wasn't going to be a long-term solution.
Right. That is the thing about the Bay Area. It is a wonderful place to live until you have to pay rent. And it's like, oh my God, this is hard.
Right. And I knew I didn't want to pay rent for the rest of my life. So, and so then it almost came full circle, right? Because I was looking around, I applied for a few positions, trying to stay in the Bay Area. Nothing was really panning out. And lo and behold, here comes a advertisement for School of Veterinary Medicine, the place where first year grad school I was going to pick up my parasites.
Oh, wow.
I knew many of the faculty there. And when I saw that position, I thought, yep, that's the next step. That's it. And so, and that's where, to bring this story full circle, that's where I would say it got really, really good. Because even at UCSF, there were some peculiar challenges there with career advancement that I wasn't aware of. There were a few instances where I felt like I was really singled out or something like that. Of course, you would know that a movie is called The Last Black Man in San Francisco. I actually went to go see that movie, and I did identify with some aspects, not all aspects, but so it's a thing, right? So I kind of felt isolated. I felt special and different, and I don't want to feel special and different. I want to feel like I feel today, which I'm talking to colleagues and sharing stories, and there's nothing else there but just our experience, right? And I didn't feel like that there. I do feel like that here at Penn.
Oh, that's great.
Since walking in the door, there's just been open arms and lots of responsibility. Hey, the professor wants you to do this. Hey, why don't you go and do that? So it's been the opposite. It's been like, hey, we really want to know what you have to say. So my calendar, it has a life of its own. I'll say that. So it's been great. And now with the number of students that have joined the lab, the different directions that we're going, it's great. I'm excited to see what happens next.
Wow. I'm
so
really.
Can you tell us a little bit about what is going on in your lab? I mean, I loved the story that you talked about it at the Upstate conference, but the connection between the nociceptors and immunology. I don't know if you want to talk about that a little bit. First of all, what are nociceptors and why are you interested in this area and connection between immunology and the neurobiology?
So let me first start by saying I did have an early interest during my postdoctoral days in South Africa. I did have an interest in neuroimmune interactions. I think, what is it? Was it Jules Verne? One of the, originators of the field of immunology, was Jules Verne, wrote about the potential interactions between the nervous system and the immune system because of how the systems are laid out. The cardiovascular system and seems like the neurons follow along where the blood vessels are, and there's just some really fundamental aspects of mammalian physiology that kind of bring these systems together. And so the implication was that they're communicating. right?
Right.
And so I was interested then, but I was too busy being an M2 macrophage biologist, right? And so shortly before the pandemic, I had a couple of rotating grad students, and then when the pandemic started, they were like, "Oh, I want to come to your lab," because there was this frantic grab for students to find laboratories, when the world was kind of coming to the end of this. So we thought, and I have all that lead in to say that three students committed to the lab at one time, and I had a postdoc coming. So the two of the grad students and the postdoc says, "Dr. Herbert, we really like your research." Sorry, it's the mail carrier.
Oh, it's fine. Yeah. I was like, is that a dog or a kid? You could bring him in the shot.
Yeah, right. No, it's my rescue dog. I'm sorry.
Oh,
okay. No worries.
He's a sweet dog. If you're not a mail carrier.
Okay.
But to get to the point, that the two students and the postdoc both said, "No, we like your program. It's great. We identify with you. These are individuals from populations of people of color. You're great. We want to work, but the only way I'm going to come if I have a neuroimmunology project."
Wow.
And I had no neuroimmunology projects in the lab. Nothing. Zero.
You're like, oh, crap.
Right. That's exactly what I said. Oh, crap. But then what came out of my mouth was, okay, no problem. Because, I wanted to grow the lab. And so to date, we have 3 really interesting and new projects that are all neuroimmunology focused. They're all led by these respective individuals, one of which we've just submitted for publication. The one that Cindy, you heard is just been submitted. That one focuses on schistosomiasis. That parasite enters through the skin, and in the skin of all of us are small endings of neurons. We call them afferent endings that reach to the uppermost layers of the skin surface. And their job, they're decorated with what are called ion channels, also bona fide receptors that bind certain ligands, but ion channels, it's like a tube, so those channels sense and respond to clearly ions, but also heat and what we call noxious stimuli, painful or hurtful or dangerous. That's all so by definition, they're called nociceptors because they respond to noxious agents.
Interesting.
Right? Something irritating on your skin, guess what? Those neurons are just firing, going crazy. So the basic question is, when these neurons in the skin are activated, are they communicating with the immune system? If they are, what is the manner or what's the mechanism? What's responsible for that? And what's the outcome? And so Juan's work shows that in the context of schistosomiasis, if these nociceptors are preemptively activated, so we can do that with a few genetic tricks of exposing them to laser light, a blue laser, so we're not harming animals, but it specifically activates those neurons. The parasites are not able to efficiently enter the skin.
Wow.
Like it's almost like a force field. Like we measure and count the number of parasites that are able to disappear and go into the body, or the ones that are still in the solution that we put on the skin. And one such a keen astute scientist, one of his first experiments was, hey, these parasites are not going to the skin.
Wow.
And so we have a whole mechanism that I won't bore you with all the details, but in essence, the neurons, as soon as they're activated, and we're still looking for a natural activating stimulus, they tell the local immune cells, hey, change your cytokine profile, get ready, because there's a pathogen coming. and repel that pathogen. I mean, neurons respond in nanoseconds, right? The immune system responds in, hours to days. The nervous system is just like immediate almost. So I think it's pretty exciting. And I hope that it gets a fair review, because we're mingling that mechanism in with the understanding of a particular interleukin called a cytokine called interleukin 33. And there are a lot of scientists across the world who are trying to figure out how IL-33 works, because this is a cytokine it's been implicated in a whole host of diseases that we do suffer from here in the U.S., like, metabolic disorders, developmental defects, cognitive defects, not to mention inflammatory bowel disease and allergic asthma. So IL-33 is serving a very central role in a lot of different areas. And I kind of jumped the gun on some of those questions that were posed here, that, and this is one of the ways how studying parasites and how they interact with mammals, or how they interact with mammalian immune system, can teach us basic immunological mechanisms that can be applied to these other types of diseases. So we're really doing like kind of basic mechanisms, how this works. And I hold that the parasite is really the ideal situation because of what we call coevolutionary adaptation. So you have two organisms and they're trying to live together, and then they kind of meet in the middle and figure it out, kind of like a marriage, you kind of battle a little bit and then come together. You're like, okay, this works for both of us. So we'll just do it this way, and that coevolutionary adaptation allows the parasite to modulate the immune system and the immune system to modulate the parasite. And so if we follow the parasite, we can discover immunology. And that's really my theory that really drives the work. So there's a skin project. There's also another skin project where we're looking in the foot pad, and Yvonne is leading that. And Yvonne has a assay where something similar happens. It's a different pathogen. It's more of a hookworm type pathogen, but she also is seeing, she's uncovering what are the mechanisms of how those worms are repelled from the skin surface. It turns out that that in humans, skin immunity doesn't develop all that well. So if you look at endemic regions across the globe, people are repeatedly infected with hookworms, even after drug treatment. Their immune system somehow can't figure out how to resist a secondary exposure. Well, in mice, resistance to a secondary exposure works really well. So that system is more about adaptive immunity. The previous project is more about the initial encounter. And so we're kind of dividing those skin projects by parasite and location. And I think we're going to find some interesting things in both. The last one is probably just my pet project, because my favorite, because not only is the student who's leading it just a joy to work with. And he's really he does so many different things. He's a very passionate person about DEI. He gives up his time to his colleagues, his fellows. The faculty love him. They're always, Jay, please help us. And Jay has established all on his own. We just talked about it theoretically. I didn't have to go to the animal colony and show him how to do anything. He's figured out how to study the sinonasal tract.
Wow.
And so, we're hopefully coming out of this pandemic, and, immune responses in the nose and in the sinuses have been historically kind of ignored by immunologists. I mean, we know that they happen, but most studies, and we've done these studies, focus on the lung. What happens when things are in the lung? Well, a lot of times, by the time, like COVID, once it goes down into your lungs, you've got serious issues to deal with. But it starts in the nose. So we devised a system where we're studying how allergic stimulation in the nose leads to a response where the neurons that are extending their afferent impulse into the airspace now, right? It was thought that only the olfactory neurons did this. So way back in your sinuses. The olfactory neurons, why smell is such a, economically feasible avenue, right?
Right.
Smell, among many other things, or how a smell can send you back. A particular smell can send you back 30 years in an instant, right? It's because those olfactory neurons are right there in the airway, and they are tied directly into the central nervous system. So you can smell, and there's a thought, period. Well, we found another set of neurons in the respiratory tract that's more in the proximal regions of the nose, that when stimulated with an allergen, so what Jay is using is fungal allergens. I mean, we probably know someone who's allergic to fungus, go down in the basement and you can't stop sneezing.
Yeah.
Right. So Jay, again, I probably should have had Jay on here today.
This is Jay Ortiz, correct?
This is Jay Ortiz.
Yeah. We follow each other on Twitter, and I've interacted with him, and he's such a he
is remarkable.
Great. I've never met him in person, but yeah, seems like a great person. That's awesome.
Down. Jay has figured out how to quantify mouse sneezing. I didn't know before Jay. I had no idea a mouse could sneeze. I didn't know they could sneeze. They could sneeze. How are you going to record that? Like, he's figured out a whole with video and with cameras and a plexiglass box and how he administers the agent.
That's amazing.
Figured out how it's pretty amazing. And what his project, and he's writing it up right now. We hope to submit it for review in a couple of weeks. And the gist of it is that the neurons that extend into the airway from the respiratory epithelium, they sense allergen. And when the neurons sense allergen, they then can produce factors, neuropeptides, that tell the immune system, make an allergic response. So if you get rid of the neurons, or if you provide the ligand that the neurons release, you can reproduce probably about 60% of the whole allergic sequelae, like all of the factors that we've known for probably 30 years that are characteristic of an allergic response, sneezing nose, itchy, watery eyes, that whole, all that physiological stuff, that makes you grab for your Kleenex, and take, ibuprofen or something. We can recapitulate by tinkering with those sensory neurons. And I think
so the $1 million question is, can you block them? Right?
Can you block the neurons, right?
Yeah, because then you could think about interrupting either development or perpetuation of the allergic diseases, right?
Please get rid of my allergies.
Right? Can you imagine? I'm just frustrated with this project because I don't know of any worm species that go into the nose. I'm talking because I would bet dollars to donuts that if you put a worm in there, that it would block the allergic response. That's what I think. But,
and how is the neuron recognizing directly the antigen? Do we know
that's
that?
what we don't know that yet.
Okay.
We don't know that. That is definitely $1 million question. Neurons, surprisingly enough, express a lot of different receptors. They do express what are called, like Cindy would know, this class of receptors called toll-like receptors. And toll-like receptors were discovered a few years ago, and there's a Nobel Prize jointly awarded about that because they are germline encoded pattern recognition receptors. So they sit on the surface of cells and they recognize anything that looks characteristically like a pathogen, right? Or things that most types of pathogens would make. So toll-like receptor 4 has been described to be expressed on sensory neurons. Toll-like receptor 4 recognizes primarily lipopolysaccharide, which is the fundamental structural wall component of gram-negative bacteria. So that means if you have TLR4 on your surface, you could sense any instance where there's gram-negative bacteria in the area. Now, that's not answering your question because you asked about allergen. allergen is just a whole other beast altogether. The understanding of how allergens are recognized is a very, very hot topic. And that's just not by neurons. That's by any cell. There have been a few papers published on it, but then, it doesn't explain the whole thing. I mean, can you imagine the same receptor that would recognize nickel versus a fungus? I mean, these are completely different things, and they drive allergic responses. So I think there's a lot to be done here. What my lab specializes in, I would say, in addition to using parasites as a model, is to uncover cellular communication mechanisms.
Right.
What really fascinates me is where you have this cell over here that's one type of cell, and you have another type of cell. How do they talk to one another? How do they influence each other's behavior? What happens if one exerts stimulus X? How exactly does that affect, cell stimulus Y, if you will, right?
So I feel like I, have only about 1 million questions, some of which are, related to a lot of questions about coevolution. But the thing that I was really excited to ask you is that in one of your publications, you mentioned this idea that every cell is an immune cell.
Yeah.
And I love that.
Yep.
And I kind of wanted to know how you think about that concept and your thoughts on that. Because I love it.
Well, it's not too glamorous, but it's an extension of what I've been saying. So the cytokines, cyto, cell, kine, kinesis, how do cells move? How do cells communicate? Are powerful agents, right? And every cell that we know of, from everything from a fibroblast to a neuron to a adipocyte to definitely immune cells to muscle cells, secretes or releases cytokines. So if a cell is damaged or infected or undergoes some malignant transformation, its communication language shifts. And that shift in the communication can then communicate to neighboring cells, maybe immune cells, to either call in immune cells or tell them to go away. If you're like tumors, right? That tumors make all these cytokines and just say, I'm not here. Don't respond to me. A lot of people think in parasites and tumors may share some really interesting similarities. And then there's the receptors, right? So how does, every cell is an immune cell because, they express a complement of, molecules on their surface that can receive those signals. So if there's a T cell that goes into the muscle of someone who's just received their COVID shot, right? The T cell or dendritic cell, they go into an area where they're not really usually there. And then they see, oh, wow, there's all this interesting foreign stuff. Let me make an immune response. And we benefit from that by being, having immunity, at least for a little while, against COVID-19. So what we're trying to do with that manuscript is kind of reshape the discussion that immunology means T cells and B cells, maybe macrophages and dendritic cells, and that's immunology, right? We're trying to say, whoa, wait, hold up, immunology is pluripotent, we're kind of robbed the phrase from that term from the developmental biologists, every cell has a capability, and as you alluded to, many different types of cell can be infected because, of course, pathogens mostly want to avoid the immune system, right? And so they probably are infecting cells like, you would normally think would have an immune cell function, neurons, if you're, a chronic viral infection like herpes virus or something like that, or, in muscle cells or adipocytes or something like that. But then those cells call in their help right? You get the communication again, right? So here's the muscle cell infected. It's calling in the army to help out. So every cell is an immune cell.
I've told my students earlier this semester that the more I understand aspects of immunology, the more I realize that actually we study the homeostasis system.
Yep.
It seems like the same kind of concept that you're talking about there.
Yes, very much so. If you will allow me to extend that, because I think it's implied in that is how parasites, the coevolutionary piece about the parasites. So one of the things I like about particularly parasitic worms so much is that in contrast to many other types of pathogens, parasitic worm infestation needs to last for a long time in one host. And that's because after they've committed so much to developing into an adult form that's reproducing, and that, we all think about, selfish being, right? Like propagating that DNA, propagating your genetic code. Well, that propagation of genetic code is dependent upon, it's kind of like one worm in, one worm out. They don't generally divide, they don't undergo asexual reproduction and can divide and, undergo budding like bacteria or viruses, make more of themselves. They don't make more of themselves inside the same host. They're relying upon their genetic material being passed out into the external environment, undergoing a development there, and then being reintroduced to a new host. Wow. So that means by definition, adult worms are going to devote, I'm being a little bit anthropomorphic here, but have devoted a lot of time and energy to ensuring that once they infect a host, that they can stay for a long time. So I'm coming back to this idea of like the correlator between a tumor and, adult worm. You don't know I'm here. Talking to the immune system, like, you didn't see anything. I'm just sitting in the intestine, or I'm just sitting in striated muscle, or I'm just sitting in your central nervous system.
Nothing to see
here. Nothing to see, right? And so, so that's the question. Of course, immediately comes: how do they do that? How do they do that? How do they pull off this magic trick? And that has fascinated me for a long time. And I'm a little sad to say that we have not led the charge on that. There's some investigators over in the UK. Rick Maisels comes to mind, who really rocked the whole.
Another mailman, maybe?
Yeah, he's just looking out the window. Rocked the whole world with the recognition that there are specific molecules released from adult hookworms that are able to tell the immune system shut down. They're able to tell the immune system turn on your internal suppressive system. So in our immune system, we have certain cells that are devoted to just tamp down immune responses, right? I mean, you can't always push on the gas. We need a break, right? And so worms have figured it out. Worms have definitely figured out, hey, you know what? Not only are we going to produce factors that tell a lot of the other immune cells just stop responding, but we're going to make a molecule that's going to tell progenitor type cells or naive T cells really to become suppressors. And not only do they tell the cells to become suppressors, those suppressors, when formed, suppress everything.
Right.
So what Rick and other colleagues have not only identified specific molecules, and yes, they're they're starting companies on this, but this has led this thought of how people should just be infected by worms if they have a chronic inflammatory condition like inflammatory bowel disease, or I've seen reports on MS, on celiac disease. Celiac actually works pretty well. There are now human challenge infections with hookworms.
Right.
Sponsored and run by the National Institutes of Health, specifically for this purpose, to block chronic infectious disease I'm using worm infections because the worms know how to do it. They've put eons of time into how to twist the immune system right at the right level, right? So you're not completely shut down. It's not like taking a big dose of corticosteroids, right? We know what like that has other negative consequences. So worms don't want to completely eliminate immunity. Many of their life cycles do depend on some degree of immune stimulation, but just not too much. So it's kind of like the Goldilocks effect. Worms like, okay, not too much, not too little, just right.
Right.
So these trials that you're talking about with worms, how do you prevent any pathogenic effects from the worm?
There are GMO worm, good GMP, good manufacturing practice worms that have been. There was. I think this professor is long retired now. He was in the UK. Was it Pritchard? David Pritchard, maybe? I forget. But he went to Papua New Guinea, self-infected himself with hookworms, came back to England, and cultured them. Removed every other, in the laboratory, removed all the other pathogens. Yeah, I know. It's
quite the commitment.
Cindy, parasitologists are a whole other breed of scientists.
I do know that. We have some here. They are a different breed. I would agree with that. Yes.
Some investigators at NIH got a hold of these worms, and they're doing challenge infections on human forearm skin.
And they're using the human pathogen, right? Because wasn't there was it IBD? Weren't they using Trichuris suis? So pig.
Right. There was another investigator
helmets for driving TH2 type of responses or something to these responses.
Yeah, that was a big splash a few years ago, actually during my postdoc days, by investigator who was a gastroenterologist, Joel Weinstock. And he had a few early successes in using Trichuris suis, which is a pig whipworm and lives in the colon. He was like a really convincing guy, very, personality as large as an auditorium. And so I guess he got, some volunteers that were ready to take whipworm eggs. And the idea was that he could easily obtain whipworm eggs from local abattoirs. It wasn't a human pathogen, so that this species would have a abbreviated infection in a human, right? It wouldn't live for, like I described, the human worms decades, right? But this would live for a few weeks. But during those few weeks, he showed the worms would hatch in the bowel, and they would then expand that T regulatory compartment. And that expansion of T regulatory cells in the early trials led to suppression of Crohn's, Crohn's disease. And it was really exciting. But then, to move it forward, well, you can't just start giving people pig parasites,
People can still buy them online though, right?
You can buy them online. But you're not.
We do not condone treating yourself with Trichuris suis.
If anyone is listening to this intently, do not take what Dr. Herbert saying like, "Oh, Dr. Herbert told me to go." No, Dr. Herbert did not tell you to do this. Okay, do not do this at home. But subsequent trials failed. So they started to use just the eggs, and they were irradiated, and various things were done to really clean them up, as you mentioned, Vincent. And then it just kind of dried up. They were not reproduced, but the notion still stands. And there's enough of subsequent reports and other systems. Like I said, I have some other colleagues who are very, very committed to this, and the data look promising. There's various other types of worms, but worm infections to cure excessive inflammatory disorders, chronic inflammatory disorders. I think we're going to see more of that.
Do they stay? So you said they do it in arm in the arm. They infect the arm. Do they stay there, or do the worms migrate? And could they take them back out if something goes wrong? I mean, like, is there?
Yeah. So the trials I read about and the investigator I know, you get a full life cycle. So let's come back to hookworm in particular. You put it on the arm. Volunteers experience a little itchiness at the infection site, perhaps just a little bit. Nothing, not getting to adverse effects, serious adverse effects. Those trials are set up where if there's abdominal discomfort a few weeks later, as the adult worms have developed, there are highly effective therapeutics out there.
Antihelminthics.
Antihelminthics that work pretty well. But like I said, the problem, of course, is that process can go over and over again, right? I mean, of course, now most people realize, oh, I've had COVID. So there's this period of time where my immune system is going to protect me. The problem with coronaviruses is that protective immunity doesn't last very long. But in hookworms, it doesn't seem like it develops at all.
That's wild.
You don't even get 6 months of, you can come back after doing national administration, and 2 months later, all the children have full complement of worms again.
Which really speaks to their ability to evolve with humans and avoid the immune system. I mean, it's incredible. And I'm curious, from a big picture, we talk a lot about the hygiene hypothesis, and that for as long as humans have been around, we've evolved with them, and now we no longer. I mean, our goal is to not have a helminth infection.
Right.
And so, I don't know. Can you speak broadly to that hypothesis? Because I think it permeates many aspects of immunology.
Yeah, this is the type of question I'd like to bring in the analogy of warfare, and how, in my opinion, and I know many others' opinion, not everyone's, however, the immune system evolved for host defense, right? Because there are constantly agents that are trying to take advantage of us and so on and so forth. Well, if that's true, that we have all these particular types of immune cells poised for host defense as a big army with all the different branches of the military, well, what happens when there's nothing to fight?
Right.
They still practice.
They're going to find something.
Go to the bar, go to
cause trouble.
Let's just shoot off a missile. Let's just shoot one, right? And so with that analogy, I think works really well. So the hygiene hypothesis loosely is based on our body needs to be, our immune system needs activity. It's going to be an active way. We make millions and millions of neutrophils a day. Many of them die. They make more. The bone marrow is constantly working. Homeostasis doesn't mean nothing's happening. It means there's a turnover. It means there's a balance between the birth rate and the death rate, and so on and so forth. Well, these cells they're designed to look for things, to respond against, to engulf, to produce cytokines, to coat with antibodies, to sequester, to eliminate, through the spleen or through, excretions, you name it. This is a machine that's constantly working and looking for something to do. So, and there's a lot of examples of this where, particularly early in life, exposure to, pet dander, pollen, you get, stuck. These are all the things from my experience in Mississippi, right? In briar patches, you've got scratches and scrapes and bruises. You're getting bitten by mosquitoes. You're getting, all these various things. It gives your immune system something to do. And what happens, just like you go to the gym, you get in shape, right? You're in balance, right? Oh, I did that, and so we have memory kind of ingrained in our immune systems as we get older. So there are certain infections that elderly just don't get. because they've had so many previous exposures. Their immune system is poised and trained and it's healthy, right? And that's why, from 0 to 5, the juvenile, I will say, or the infant immune system, the immune system that's in us as we are very, very small children, is really ignorant of the world and can be really taken advantage of by pathogens. That's why, death in that range is such a big problem. It's still a big problem, particularly in, low and middle income countries. And so we need those exposures. We need to be on a farm, exposed to, stepping in, cow manure, or, getting a bunch of hay dumped on our heads, or swimming in a lake that has, I don't know, something. Hopefully you don't get amoeba, you won't survive that. But you know what I mean? The exposures early in life train our immune system to be healthy, right? So we don't develop problems down the road. In its absence, and you see this in a big way in places like Japan, definitely here in the U.S., the westernized countries, but all over. Now that we live in environments that for sure are devoid of worms, for sure, right? But also can be so clean that the immune system doesn't even see, your child's not being licked in the face by the puppy that just went outside and ate something off the ground. When that doesn't happen, you've got an immune system that once it gets to a certain age is now hungry, looking for anything to act on. So hygiene hypothesis states that now that's why, to answer Brianne's question about this maladaptive response. So instead of fighting a worm, instead of fighting, I don't know, a tick bite or something like that, the immune system now sees, pollen, that's dangerous. We need to make a response against that. But that is just
that's the worst thing we've ever seen, right?
That is the worst thing we've ever seen.
Right.
So that's the idea behind it. And like any hypothesis, there's a lot of information, a lot of evidence that supports it. And there's a fair amount of evidence that refutes it. And so we're trying to figure it out, right? What's the real truth there? And how can we potentially manipulate it to block some of these diseases? And like you said, please cure me of my allergies, and honestly, are the current therapy, the best that we have right now is either a therapeutic that blocks the end stage, like, okay, so we can block something that will stop your mucus. Like you still have an allergic response, but, your friends will not run away from you. We can stop the end, or we will give you the thing that you're allergic to in small doses,
right?
Over and over and over for a long time. And we're going to slightly increase the dose until your body realizes, hey, you know what? We can just stop responding to this. So that's immunotherapy, not immunotherapy. It's more like intolerance, where T regulatory cells, our body's suppressors, will then start to be specific for this. So it'll say, okay, when this antigen comes along, we'll suppress a response. We won't, make one. But it's risky. It's time intensive. It's costly.
Sure.
We definitely need something better than immunotherapy. And what about people who are allergic to 6 different things?
Right. That would be very challenging to do all of those different.
Yeah.
Right.
They do mixtures.
It's potentially life-threatening. It can be like anaphylactic shock can kill you very fast.
Right.
If you're exposed, people can pass away from a wasp sting. The venom that's in certain wasp species, or bee venom, can kill someone very rapidly. So it's serious. It's serious.
And the funny, not funny thing is we're covering this in the course that I'm teaching now. And we were having a conversation yesterday, and we're talking about allergic immunotherapy. So this tolerizing thing. There are a couple of people in the room who have done it. And they're like, yeah, you do it in a doctor's office. And then the next time you could do it at home, but they send you with an EpiPen because sometimes you have a reaction.
Yeah.
But they said both of them have gone to the doctor to have their shots and for whatever reason had an adverse reaction and passed out and had to be revived with EpiPen and go to the hospital.
Wow.
So it is not, people are like, oh, I'll just take allergy shot. It's not that straightforward. It is, there are very significant risks associated with that. So there's a lot to be gained from developing other ways to control allergic responses. So it seems like maybe your stuff could lead us there eventually.
Yeah, maybe so.
Some evidence.
Yeah, like just spend the rest of my career trying to figure it out. I do want to give a nod to an immunologist who I admire tremendously. She's a controversial figure, but Polly Matzinger.
Yes.
I once saw her. I saw her twice give a talk, and I was just mesmerized at both times. Yep. She came up with a danger hypothesis, which states that just like what I mentioned about toll-like receptors, which there's molecules that look like pathogen. So the danger hypothesis was a little bit different in that it's anything that's dangerous. So back to nociceptors and noxious stimuli, anything that the body thinks is injurious, right? So this can be chemicals or whatever, just blunt trauma, right? That's dangerous. We don't want blunt trauma. But those things can also drive immune responses. And in the, I think it was the New York Academy of Sciences where I saw her, she challenged everyone in the room to refute that hypothesis, and that she said she could explain all of immunology with this danger hypothesis except allergic responses. You pointed that out. She's like, well, okay, allergic responses, I don't know. Let's not talk about it. It doesn't explain that. So that's why I bring up this example that there's something about the allergic response that it's just not intuitive. It just doesn't make any sense to like, how can I be allergic to nickel? What is it about nickel that will cause my ear to fall off, like from an allergic response? It's just, and how does that have anything to do with, pet dander or pollen or grass? I mean, it's on and on and on. There's so many things that humans can be allergic to. Milk, food. It's hard to get your head around it sometimes.
Let me know if you need a test subject.
So I had a question that kind of goes back to the first part of your talk, and I want to make sure we don't end your science piece, because it's just been wonderful to hear about your career and the science you do, and it's so clearly important. But I think to go back, it was such an honor to hear your story. I mean, that I'm glad you went into the length you did to tell us from start to finish, because there's just a lot of lessons in there. And I'm curious, looking at today, we just ended the week of Black in Immunology. I think November 13th to the 19th, they had a week where Black immunologists could speak on their science and science communication. So what do you see today as science, DEI, being a Black scientist? What do you see hope? Are you seeing change? And can you speak to the changes you've seen and what gives you hope for the future?
I think, as you were posing your question, the word opportunity just flashes in my mind. I think there is endless opportunity today, doors that are open, minds that are open to considering, right? That the proposed link between a person's features versus a person's contribution has nothing, like they're not connected. They're not connected to, if I'm this, then I can only do that, or I'm going to do this type of science or whatever. I think that there's equal opportunity now for this discussion to be had about reaching your potential in biomedical research. I'll speak to, Kizzmekia Corbett is someone who I admire tremendously. I mean, when I would speak to, in my social circles, particularly in the area of religion and so on and so forth, the Black church is a very prominent force in the country. And, because of the past atrocities that have been committed against people of color for, medical trials and things like that. Of course, I don't need to go into that, but we know that it happened multiple times. There are a lot of, insecurity and like suspicion, right? Mm-hmm. That COVID vaccines were not another attempt to do something untoward, right? And I think an example of a Kizzmekia Corbett smashes that, right? Here's someone who been devoted their early part of their career and now is just, I think she's just getting started, actually, to make a really fundamental contribution that helps all of us, right? And so that was really powerful for me to be able to go to a congregation and say, hey, well, I would have you to know that the person instrumental in devising this COVID vaccine was someone with African-American descent, someone looked just like you. So the chances of this person, devising something that's going to hurt you or that's not beneficial, it's not to your benefit, is very, very slim to none. So that was one of my talking points. It still is. So I think we are in emerging state. I mean, of course, resource allocation is always going to be contentious, right? Congress only gives so many dollars to, so there's always going to be a balance on how monies are being spent. But just, places like the HHMI who are creating, possibilities for people of color to secure their early careers. When you're, like I said, like in infants, right? That early stage of your career is really make it or break it, right? It's really so essential to get the right support around you in those early careers. I think there's a big difference from like my time, right? 80s, 90s, that type of thing, when it was okay for someone who was a mentor or superior to say, oh, no, you know what, why are you going to do this? I think you should do this. And that be a defining factor in shaping someone's career to now. Right now, not only do I think, that type of statement would be really give pause, telling someone, oh, no, I don't see you going in this direction. I think you should do this, I think, I'm sure it's probably said in some circles and some conversations, but I think it's less likely. I think trainees are starting to see, they can envision themselves. And because what I've been hearing mostly, I mean, the number of invitations that I've gotten, and that there's a group of students saying, how did you do it? Like, really, can I, imposter syndrome, all this kind of stuff, I didn't even know there was a name for that thing, by the way, I said, well, we look at you and say, well, gosh, if you can do it, I can't. I surely can, that has been so inspiring to me, and that what further drives me forward, to do the best science that we can possibly do. So I think it's opportunity now. I think there's a lot of ample opportunity. People still need to know that, and I do want to speak to this, just because you have a certain demographic, I think there is a large sentiment that's like, well, we're not going to lower our standards or lower rigor or lower any kind of, just to, be more inclusive. And I don't think anyone wants that. Honestly, I don't want it. Right. I just want equal opportunity. You have people who have equal potential. We'll give them equal consideration. And I think that's the conversation that's been happening. That's where we are now. I'm going to continue to believe that, I think,
I think the view of Kizzmekia's work that you just presented is, I've never thought of it that way, but it's perfect. I love that very much.
Yeah.
I think that people need to hear that more, right? I think that's great.
Yeah. I need to know.
Yeah.
Absolutely.
Well, we have kept you long enough. So
I know. This was awesome.
Been wonderful.
Let's wrap this episode
amazing.
62 of Immune. You can find show notes at microbe.tv/immune. You can send us questions or comments, immune@microbe.tv. If you like our work with all of these science programs, consider supporting us. We do it for you, and we don't use ads or anything, so we'd love your contributions. Go to microbe.tv/contribute. Our guest today from the University of Pennsylvania, De'Broski Herbert. Thanks so much. It was great to hear your story and your science today.
It was truly a pleasure. I had a lot of fun doing it.
Cindy Leifer's at Cornell University. Now, Cindy, you want to give your Mastodon handle now?
It's the same as my Twitter because I'm lazy, but yeah, I'm both at Cindy Leifer on Twitter, and I did do a Mastodon at Cindy Leifer at med-mastodon.com.
All right. Thanks, Cindy.
We'll see if it takes off.
Steph Langle's at Case Western Reserve University. Stephanie Langle on Twitter. Thanks, Steph.
Yeah, thank you. This has been great.
Brianne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thanks. This was really fascinating. I learned a lot.
I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.